Now, you see here in this slide is a red blood cell membrane, a photoelectron micrograph of a red blood cell membrane surface. Now, what's distinct or peculiar on this picture is that there are actually pores or holes found on the surface of this red blood cell. Now, normally, you don't get to see this, no pore formations. But what has caused this was that the complement proteins, upon binding to the surface of this red blood cell membrane, it has caused these pore formations causing lysis of the red cell. So therefore, when these complement proteins become activated and when they bound to a cell surface, the end result of complement activation is cell lysis. Now, I will be explaining in detail what happened here in the next few slides. Now, these are the objectives for today's topic. Now, there was a scientist named Jules Verdet in 1890s who conducted an experiment. So what was that experiment conducted by Jules Verdet? So he used an anti-serum, and this anti-serum now is specific to the bacterium Vibrio cholerae. So remember, in your first few topics lectured by Miss Lorraine, you learned that we have this what we call antigen, and we also have this what we call antibodies. Now, your antigen basically is more or less contained in the surface of anything that is foreign or not considered by your immune system as self. So they are actually antigens, but your antibodies, on the other hand, are actually produced by your immune system, which is specific for that particular antigen. So what happens is that your antibodies contained in your serum would actually help get rid, will neutralize those antigens found on the surface of foreign substances. So what happened in the experiment of Jules Verdet was that he added an anti-serum, he isolated an anti-serum from the sheep, and this anti-serum contains antibodies now specific to bacterium Vibrio cholerae. So specific to the bacterium Vibrio cholerae. So what do you expect? Since these antibodies are specific to the antigens found in the Vibrio cholerae, you learned that these antibodies in the previous lectures would actually get rid of those antigens. And so what happened was that it causes lysis of the bacteria or the Vibrio cholerae. So again, sheep anti-serum added to the bacterium Vibrio cholerae, it causes lysis of the bacteria, the Vibrio cholerae. Now, Jules Verdet was actually not contented with that. So he further heated the mixture. So the mixture originally containing the sheep anti-serum and the Vibrio cholerae, he heated it at 52 degrees centigrade. And guess what happened? The serum now, the anti-serum now, was not able to lyse the Vibrio cholerae. In other words, heating the anti-serum destroyed its bacteriolytic activity. So you might be thinking of something else now. How come that the anti-serum lost its bacterial activity after heating? So again, being a scientist, curious as he is, Jules Verdet further did some manipulations or treatment to his experiment. So what he did was the original mixture containing the anti-serum plus the Vibrio cholerae, he heated it, no lysis was found. So what he did was he added another volume of fresh anti-serum. Fresh anti-serum containing again antibodies which was able to kill the Vibrio cholerae. So what happened after addition of fresh additional serum? There was now restoration of the bacteriolytic activity. So again, Ship antiserum plus Vibrio cholerae equals lysis of bacteria. But when Jules Bordet heated the mixture, there was no lysis of bacteria. But after adding another volume of your fresh normal serum, this actually restored the bacteriolytic activity. So, question, if you were Bordet, what would you hypothesize? Okay, so I'll give you 10 seconds to think. Okay, so I hope we have the same answers. These are actually the conclusion of that activity. Bacteriolytic activity requires two different substances. The specific antibacterial antibodies, which are resistant to the heating process, and a heat-sensitive component responsible for the lytic activity. Now let me explain these things in detail. Okay, so let's start with letter A. It says here, bacteriolytic activity requires two substances. So number one, specific antibacterial antibodies. So remember in the experiment of Jules Bordet, what happened was that he added an anti-serum containing antibodies specific to Vibrio cholerae. And these antibodies now has caused lysis of the Vibrio cholerae. So it killed the Vibrio cholerae colonies. So in other words, for us to produce a bacteriolytic activity or to kill these organisms, we need antibacterial antibodies. And upon heating now, these antibodies are actually resistant to the heating process. So if whatever heating you do, your antibodies now will still be found in your serum. But there are actually heat-sensitive components responsible for the lytic activity. So remember, after heating, what happened? Correct. What happened was that there was no more lysis of your Vibrio cholerae cells. But after adding fresh, you added fresh anti-serum, what happened was there was lysis. In other words, originally, there were components in your anti-serum that was used by Bordet which was destroyed by the heating process. Thus, the heat-sensitive component also is responsible for the lytic activity since upon addition of another fresh volume of a serum, what happened was there was lysis. Okay. So later they call these heat-sensitive component as the complement system, which was, when heated at 56 degrees centigrade for 30 minutes, has caused inactivation or destruction of the complement components. So with that experiment, Jules Bordet was awarded Nobel Prize in 1919 for his role in elucidating the nature of the complement. And during that time, Jules Bordet did not know the name of this heat-sensitive component. It was in the experiments of Paul Ehrlich, to which he termed these proteins now as complement system. Why complement? Because it supports, it supplements, it complements the action of your antibodies. In other words, it enhances the effect of your antibodies. Pellemer and colleagues in 1950s discovered an antibody-independent pathway which they termed as classical pathway. So later I will discuss that in detail. So the complement system is actually a series of more than 30 soluble and cell-bound proteins that interact in a very specific way to enhance host defense mechanisms against foreign cells. Now, why was the term complex series, no? Complex series of more than 30 soluble proteins used here? Because complex, it's because meaning there are actually many proteins that make up the complement system. And how many of those proteins make up the complement system? More than 30. So there is that term here used, more than 30 soluble and cell-bound proteins. In other words, your complement system or the proteins that make up your complement system might be soluble and cell-bound. The term soluble here would actually mean that they are present in the circulation. Soluble, no? They are present in the circulation. Whereas the term cell-bound here means that there are proteins that make up the complement system which are bound to surface of your cells in the immune system. So again, I will repeat. So we have 30, more than 30 proteins comprising your complement system. And of these 30, more than 30 proteins, we have two subsets, soluble complement proteins and cell-bound complement proteins. Soluble, it's because they are found in the circulation. Cell-bound, bound, meaning they are attached to surface of your immune cells. So these proteins interact in a very specific way to enhance host defense mechanisms against foreign cells. Now, your main goal, no, the main goal of your complement system is to actually act, no, enhance, no. That's why it's termed as complement, diba? Complement is because it complements, it supplements the effect of your antibodies. It enhances the host defense mechanism against foreign cells. Now, the complement system constitutes 5% of the serum globulin fraction. So it's a globulin, your complement. And these are actually heat labile proteins. Question: at what temperature and how many minutes will cause inactivation or destruction of your complement proteins? Correct. 56 degrees centigrade in 30-minute duration. Going on to the next, moving on to the next bullet here, it says biological activities of this system, the complement system, affects both the innate and acquired immunity. Why both innate and acquired immunity? It's because there are actually three pathways of your complement system. So I will discuss this in detail in the next few slides. But for now, I want you to know that there are three pathways of the complement system. The first pathway, we have the classical, alternative, and lectin pathway. Your lectin pathway and alternative pathway, it affects the innate immune system, whereas your classical pathway affects the acquired immunity. Why? It's because we need antibodies. We need antibodies to actually cause activation of your classical pathway of your complement system. So the complement system is comprised of circulating proteins. How many circulating proteins and what are these subsets again? Correct. We have more than 30 complement proteins. What are the subsets? Soluble and cell-bound complement proteins that promote inflammation and host defense. So the existence of this system of proteins was inferred late in 19th century when it became clear that fresh serum had the ability to lyse gram-negative bacteria and cholerae vibrios in the presence of specific antibodies. So this was actually the experiment done by Jules Bordet. And the main function of your complement system is to actually enhance, no? Enhance the work of your antibodies or innate or acquired immune system to destroy the microorganisms. Now, I want you to look at the third bullet here. It says it promotes opsonization and lysis of foreign cells and immune complexes. Chronic activation can lead to inflammation and tissue damage. So it says it promotes opsonization. So again, what is opsonization? So I think I don't need to define that, no, because you had that lecture with the Miss Lorraine, I think. So opsonization is actually engulfing, no, or removal of foreign bodies by your neutrophils, by your phagocytes. Okay. And lysis of foreign cells. So I told you already that the end result of the activation of your complement system is lysis of your cells. So class, listen here. If you do not understand these subsequent slides, you will never really pass the examination. So I want you to stop writing there and leave whatever you're doing there, whatever distractions you have there, because I know you're listening at home. And I want you to give your 100% focus on these succeeding slides. It's because this will be the foundation of on how, no, I will teach you, this will be the basics on how really the complement system works in defending your body against foreign substances. So let's discuss now the characteristics of your complement system. So this is consists of serum and cell surface proteins that interact with one another and with other molecules of the immune system in a highly regulated manner. So I am actually that type of teacher which who explains terms, no, one by one, because I don't want you to go home empty-handed and not having anything retained in your brain cells there. So at least, no, you will learn something from me. You will remember me because I have taught you something new. All right, so let me explain this in detail. Consists of serum and cell surface proteins. Again, remember your complement system is comprised of how many proteins again? More than 30 proteins. And these proteins may be, what are the two subsets? Maybe soluble or cell-bound. Therefore, the serum, the word serum here actually refers to the soluble complement proteins, whereas the cell surface proteins refers to the cell-bound complement proteins. I hope you get that. They interact with one another and with other molecules of the immune system in a highly regulated manner. Now, that might be too much for you to absorb. Let me rephrase it in simpler terms. These proteins interact in a highly regulated manner. How about that, no? More easier. So hit the more easier, no? They interact in a highly regulated manner. Now, if you are curious enough, you might ask, why, doc? Why do they interact in a highly regulated manner? It's because there is a proper sequence, a proper order to which these complement proteins, both the serum and the cell surface proteins, interact with each other. Just like when you count numbers, one comes first, followed by 2, 3, 4, 5, 6, 7, and so on. Just like when you sing ABC, A should be first, followed by B, C, D, and so on. It is the same with your complement system. Activation or activity of each complement system is highly regulated in a sense that it follows a sequential manner. So I want to direct your attention to letter B. It involves now the sequential proteolysis of proteins to generate enzymes with proteolytic activity. All right, so let's level up. Let's increase the difficulty of this discussion now. No, I'm getting more hyped now. I'm getting more excited to break down these terms for you guys. So involves sequential proteolysis. So let's start with sequential. The term sequential would actually mean in order. Kindly repeat that. Everyone say in order. In order, it's because one protein must be activated before the next protein must be activated, and then another protein would be activated next. So it follows a sequential manner. And what is that sequence? I will tell you later. So for now, I want you to remember that these proteins are activated in a sequential manner. Next, proteolysis. Sequential proteolysis of proteins. Okay. Now, your complement system are actually zymogens. You learn from your biology classes, no? That zymogens are actually enzymes which are inactive. So normally your complement proteins are inactive, no? So those proteins in the circulation and those proteins that are cell bound are actually inactive. But for us to generate an immune response for these complement proteins to perform its function, no, that is to destroy invading antigens or foreign cells, it has to be activated. And how do we activate these zymogens now? By the process of proteolysis. Proteolysis, from the word proteo meaning proteins, lysis meaning breakdown. So we from the zymogen, the bigger inactive cell, the zymogen, we break it up. We break it up through the process of proteolysis, and from that, it causes now activation of your complement proteins, and they are now ready to mingle. No, they are now ready. They are now primed to perform their function. Proteolysis of proteins to generate enzymes with proteolytic activity. So proteins in active zymogens undergoing a process of proteolysis. So we break them up, and then it causes now production or generation of enzymes with proteolytic activity, which causes destruction or lysis of your antigens or foreign cells. Letter C. So we have zymogens, di ba? I told you zymogens, and then we need them to undergo proteolysis. And after proteolysis, they are now primed. They can now perform their function in eliminating the antigen or foreign immune cells. And how do they do it? So those colored blue. So these are actually the functions of your immune system when they are activated. So number one, it causes lysis of cells, bacteria, and viruses. And you know now, you know already now from the first slide that I have shown how lysis looks under an electron microscope. And then this also causes opsonization. And this causes triggering of inflammation and secretion of immunoregulatory molecules. This also causes immune clearance. Immune clearance meaning removal of antigens, removal of antigen antibody complexes, or removal of the materials no, produced after an immune response, and anaphylactic activity. So I want to direct your attention from the last arrow there. It says there, anaphylactic activity. That is actually one of the side effects of your complement system. And later you will know that there are many side effects of the activation of your complement system. But I will teach you a hack, no, how our body, no, actually evades these side effects. So again, lysis, opsonization, inflammatory response, and immune clearance. Uh, Moving on, we have here letter D. So guys, focus. Read with me letter D. The products of complement activation become covalently attached to microbial cell surfaces or to antibodies bound to microbes and to other antigens. Thank you for reading with me. Let me explain that in simpler terms. So remember they are zymogens. After proteolysis, what happens? They are now active. No, they are proteolized or separated or broken down into several fragments of proteins. Now, once broken down after proteolysis, what happens? They are now active, they are now primed, they can participate in immune reaction. Now, these fragments, the complement fragments that are products of the proteolytic process, actually attaches to microbial cell surfaces. Okay, let me explain that in detail. So, supposing this is your microbial cell, take note. And this is the zymogen, the complement zymogen. Once the complement has detected, no, I'm ready for fight. Okay. Once the complement system has detected that, oh, there is an antigen, I must get rid of this. There is a bacteria I must protect this human because I love this human. I want him, I want her to survive. It undergoes proteolysis. And what happens in proteolysis? The complement system, for it to be activated, becomes torn into fragments. These fragments now or complement protein products which are active, which have enzymatic activity, attaches themselves. They are bound covalently to the surface of microbial cells or to the antigens. And bound, once now bound, what happens? What's the end result? It causes lysis of your foreign cells. Complement activation is inhibited by regulatory proteins that are present on normal host cells and absent from microbes. Okay, so let me explain this in simpler terms. You know, class, I'm really animated. No, it would have been better if we had this face to face. But nevertheless, I'll really try my best so that you will understand the lecture easily. All right, let's continue. There is what we call here regulatory proteins. And where can we find these regulatory proteins? On the normal host cells. Are they found in the surface of microbes, bacteria? No, they're absent in the surface of microbes. What does that mean? Listen here. Remember, you have more than 30 complement proteins. And what are the subsets of your complement proteins. We have soluble or those found in the serum freely floating as zymogens. And do not forget the second subset of complement proteins. Those found or those attached or bound to the surface of your own normal cells. You see now you see where I'm getting at? Those complement proteins which are soluble or circulating as zymogens are actually the proteins responsible for lysis of your microbial cells. While those complement proteins which are bound to the surface of your own cells are actually those proteins which controls, which inhibits the activity of your zymogens. Why? Why is there inhibition? Why should there be regulation of the activity of your complement proteins? It's because we don't want your own cells to be lysed by the complement system. Otherwise, you will die, no? Your cells will lyse by your own doing, by the doing of your own immune system. We don't want that to happen. That's why your immune system does this very good job of determining host from non-host, such that now its way of determining that this cell now is your own cell or host cell is that it has on its surface attached to it bound complement proteins which inhibits activation, which inhibits activity of your complement system. So sir, doc, ano lagi nga inhibit man siya? So we don't want too much. Otherwise, your complement system will destroy your own cells. So class, in autoimmune diseases, no, what happens is that there is loss of these regulatory proteins or cell-bound complement proteins, such that if we lost these proteins now, what happens? Your complement system will attack your own cells. They will attack your own cells and will cause lysis or destruction of your own cells. And we don't want that to happen. Are you still there? Sige, let's proceed. We're not halfway yet. We're still 5% of the entire lecture. Okay. 90% of your complement proteins are synthesized in the liver and are considered acute phase proteins. No? 90% in the liver and they are considered acute phase proteins. Why acute phase proteins? It's because acute phase meaning inflammation. During an inflammatory process, you see an increased level of your complement proteins. That's why we also call this as acute phase proteins, or those proteins which increases level or increases in number during an inflammatory process. So the hepatocyte or found in the liver produces majority of your complement proteins with exception of your C1q, factor D, properdin, and C7. So your C1 is actually produced in your intestinal epithelial cells, whereas your factor D is produced in your adipocytes. Your properdin and C7 are produced in your monocytes and macrophages. Okay. So monocytes, also macrophages and astrocytes, I've mentioned this already, that they also has the ability to produce your complement proteins. And there has been recent research showing synthesis of your complement proteins in the retina. So your eyes also has complement components there, specifically in the retina. So your complement proteins are inactivated by heating at 56 degrees centigrade for 30 minutes. So this was demonstrated by the experiment of who? Very good. Jules Verdet. Correct. So it's a non-specific serologic agent. All right, let me explain that again. Why non-specific? No, how come non-specific? Why not specific? Okay, it's because remember you have specific and non-specific components of your immune system. The specific would actually mean they are targeted. They are directed to a certain antigen. Whereas your non-specific, no, non-specific immune components are actually those targeted, or they do not actually discriminate any antigens. They may target anyone, any foreign invaders, any foreign cells or any antigens. That's why they are called non-specific, because they are not specific for one only, for one antigen, but for all. No, they target a broad range of antigens. I mentioned this already, that the components of your immune system are zymogens or pro-zymogens, meaning they are inactive until what happens? There is proteolysis. Again, question, what complement protein subset are considered as zymogens, or those, okay, zymogens, correct. Those who are soluble, soluble components, or the serum complement proteins. And the reaction starts with an enzymatic cascade. Question, the complement proteins are activated in what manner? In a highly regulated manner. Why a highly regulated manner? Huh? Correct. Because it follows a cascade or a particular sequence of activation. And I will discuss that in detail later. What is that cascade? What is that sequence? So before we move on, I want you to know that complement proteins were actually not named after a particular person, or after a particular some, or or to anyone, no? It is not named just by the thought of an author. Uh, they were actually named based on a particular nomenclature. So we have to follow a certain naming process of these complement proteins. Now I mentioned that the complement system is divided system meaning it's a group, no? System, it's a collective term, collective group wherein um, many proteins make up that system. Now, your complement system may be activated via the classical pathway, via the alternative pathway, or via the mannose binding lectin or MBL pathway. Class, I will make this more sabotable, no? Easier to comprehend to you. It's just like this. Imagine SM as the complement system. Imagine SM as the complement system. In going to SM, there are actually many routes to get there, to get to SM. You may, number one, from Mandawi City, you may cross, no? You may go along the North Reclamation Road. So that's one way. That's one route, one pathway to get to SM. Another pathway is when you were from Cebu, no, from Cebu, you pass Per Tres, and then Pier Dos, and then Pier Uno, and then you go to Robinsons, and then eventually you get to SM. That's another route. And or another pathway in getting to SM. Now, another is when you were from Ayala, no? And then from Ayala, you go to Mabolo, and then from Mabolo, the Andox there, Andox, and then eventually you get to SM. So you get my point? It's just the same with, I'm not saying that complement system is SF. What I'm trying to point out here is that there are many ways to activate your complement system. It's either via the classical pathway, the alternative pathway, or by the mannose binding lectin pathway. And this will be the lecture. The 2 meeting lecture will be about these pathways. All right, let's continue. For historical reasons, the 9 proteins of the classical pathway are designated by the uppercase letter C, followed by the number that relates to their order of appearance in the reaction sequence. All right, so let's talk about first these 9 proteins. These 9 proteins are actually proteins that make up the classical pathway. Again, your classical pathway is made up of 9 different complement proteins. And, of course, no, ato dyan ang panganlan. How do we call them? Otherwise, it will be difficult for you, no, to answer the examination if we do not know these names. So, their apelido is letter C. The letter C there, C, stands for C for classical. Capital, ha? Capital letter C. If small letter, wrong. I'll mark it wrong in the exam. So capital letter C, meaning classical. Followed by numbers. So number 1, number 2, number 3, 4, 5, until 9. So we have what are, if this is the question, what are the complement proteins that make up the classical pathway? We have C1, C2, C3, 4, 5, 6, 7, C8, C9. You get that? Very easy, no? But here's the trick here. Here's the twist. They are named in order of their appearance in the reaction sequence. Except, take note, C1, meaning it appears first. It is activated first in the sequence. Okay, C1, good first, C1. Followed by C2, wrong. This is the exception now. C1 is followed by C4, followed by C2, followed by C3, followed by C4, C5, C6, C7, C8, and C9. So, okay, why? Because C2 was first discovered, no? Why? How come now C4 goes next, di ba? Goes next to C1. You told us, Doc, nga the order of they are arranged in according to their order of appearance in the reaction sequence. So C1 must originally follow C2. Again, there is an exception to that rule. It's because C2 was first discovered and complement proteins were actually named no, in their order of discovery before their function was known to us. So, supposedly, dapat ang C4 na C2 ta siya. But C2 was discovered first before C4. But they learned through several experimentations that C4 is activated before C2 and C3. That's why the order of, if this is the question, take note, ha? This always comes out in the board examination. What is the order of sequence of activation? Activation of the classical complement proteins. Activation, C1, C4, 2, 3, 5, 6, 7, 8, 9. But if the question says, what is the order of discovery of classical complement proteins? C1, C2, 3, 4, 5, 6, 7, 8, 9. I hope you're getting that. Let's continue. Molecules that are part of the C1 complex are termed C1Q, C1R, and C1S. I want you to look at the phrase C1 complex in the first bullet. Why was it called complex? From the term itself, complex, it's not simple, no? Why? Complex, it's because it is made up of several subunits or several proteins. In this case, 3 proteins, C1Q, C1R, and C1S. And these proteins now are stabilized, combined to make up your C1 complex. So in the examination, if you just answer C1, I will mark it wrong. It's because C1 is actually made up of 3 subset of proteins, Q, R, and S. That's why its complete term is C1 complex. Moving on. Proteins reacting solely in the alternative pathway are called factors and are referred to by an uppercase letter. How do you name the proteins in the classical pathway? Starts with letter C, followed capital letter C, followed by a number. But it's different in your alternative pathway. Why? Because we use the term factor. Factor followed by the uppercase letter. For example, factor D. Or another example, factor B. So there is that term factor, followed by uppercase letter. So memorize, ha. Okay. Read with me this very important bullet here. Because if you do not understand this part here, you will have difficulty in understanding the subsequent slides. Read with me. Go. Fragments of complement proteins resulting from proteolytic cleavage are designated by an appended lower letter where A represents the smaller fragment and B represents the larger fragment. Class, listen here. You see, I'm that type of teacher which actually involves the student during my lecture. It's because you learn more, you learn best. No, there is actually that study in journals. No, that you memorize more, you remember well, you remember better your lectures or some things if you actually use more of your senses. See, you do not just listen, you take down notes, and then you participate in my discussion. Okay. Remember that there are actually complement proteins which undergoes proteolysis or proteolytic cleavage. And what do you call these proteins again? What is that? What are these specific components of the complement system? Correct. They are the soluble, or serum complement proteins. Now, they circulate as zymogens, meaning they are inactive. But For them to be activated, they undergo proteolytic cleavage or proteolysis. Proteolysis, protein slices, breaking down. Proteolytic cleavage, so they are cleaved to become activated. And these now, when they are cleaved now, they produce these parts now, these cleaved parts now, they are called complement fragments. And we also have a way of naming these fragments. For example, we have A and B. Okay, I'll give you an example. C4. C4, C4 is a zymogen. But if C4 undergoes proteolytic cleavage, C4, what happens? It produces fragments. So what are the fragments? C4A, denoting that that is the smaller fragment. C4B, denoting that that is the larger fragment. So I hope you're getting it now. C2, let's try C2. When it goes proteolysis, what are the fragments produced? I'll give you 5 seconds to answer. Correct. C2A and C2B. But, take note, huh? A represents the smaller fragment. B represents the larger fragment. Doc, how come there must be distinction of the smaller and the larger fragment? It's because the larger fragment are the complement proteins which participates in the cascade. Again, I'd like to repeat that. The larger fragments are those proteins which participates in the cascade. In other words, the smaller fragments are actually just byproducts of the activation of your complement proteins. Let's have a short exercise here. C4, cleaved proteolysis produces C4B, C4A. Which of these fragments is larger fragment? C4B, correct. Which fragment participates in the complement activation? C4B. Which fragment is a byproduct of the complement activation? Correct. C4A. Another example, C3, C3, undergoing proteolysis. What are the fragments produced? Correct. C3b, C3a. Which of these fragments is considered smaller fragment? C3a. Larger fragment? C3b. Which participates in the reaction? The larger fragment, which is C3b. Whereas the byproduct is the smaller fragment, which is C3a. But there is an exception. And what is that exception? Have you read? Okay. The exception is the complement protein C2. Why? When C2 becomes cleaved or undergoes proteolysis or proteolytic cleavage, it produces C2A and C2B. A now, C2A is the larger fragment, whereas C2B is the smaller fragment. So which of these now two fragments, C2A, C2B, participates in the complement activation process? 5 seconds to answer. The larger fragment, which is C2A. Again, that is the exception. Another exception. Another exception. C6, C7, C8, C9 do not undergo proteolytic cleavage, meaning they participate in the cascade as is without being broken into fragments like your C4, 2, 3, 5. Okay? Sige. Further degradation fragments of the large complement fragment of one components are designated by lower cases. So I mentioned that already, no? The lower case. Components that have lost activity are inactive and are usually designated by a prefix lowercase I. Prefix meaning before. So for example, C4B. C4B in itself, meaning it has enzymatic activity. But if we place letter I before letter C, making it I, small letter I, C4B, it means that that C4 is not active anymore, meaning it has lost its enzymatic activity. Very easy, right? Polypeptide chains of native complement proteins are designated by Greek lowercase letters alpha, beta, and gamma with the exception of C1q. These chains are termed A, B, C. So what I want you to remember here is that your C1q is comprised of polypeptide chains. No? Term as ABC. So let me make this simpler or easier for you guys. Diba proteins? Proteins are made up of peptides. Diba in your biology, anatomy, physiology class, proteins made up of, when you break down proteins, you form peptides. When you break down peptides, you form amino acids, no? Amino acids. So proteins are made up of peptides, no? Many peptides. That's why we term it as polypeptide chains. Now, all complement proteins are polypeptide chains that make up a certain complement protein are termed as alpha, beta, and gamma, except for C1q. We term these polypeptides as A, B, and C. Single components or multi-component complexes that have enzymatic activity are designated by a bar over the components. You know what a bar is? You know what an underline is. So underline, for example, the word here, nomenclature, if we place a line below nomenclature, line below, we underline the word nomenclature. But if we place a line above, no, above the word nomenclature, we place a bar. So it's just the same. If you see complement components with a bar on top of the name, it means that that complement protein is active or it has enzymatic activity. Active meaning it has enzymatic activity. Having an enzymatic activity would mean it is participating in the cascade. Having participation in the cascade would mean it is what? The larger or the smaller fragment? The larger fragment. See, A or B? B. Except for C2. Diba? Correlate. Proteins of the MBL pathway are designated by abbreviations of the names of the protein. And regulatory proteins are designated by a descriptive title or letter. So let's start with the first bullet, no? Abbreviations of the name of the proteins. So we have actually three proteins who are major participants of your MBL pathway. So we have MBL, abbreviations MBL, mannose binding lectin. So we have MBL1, MBL2, MBL3. Diba? So dapat na yung word ni MBL, so abbreviations. You don't need to spell out the words MBL, the words mannose binding lectin. So you use abbreviation. So the proteins participating in MBL are, in mannose binding lectin pathway are MBL1, MBL2, and MBL3. Abbreviated siya. Whereas, regulatory proteins, again, what is the other term of these regulatory proteins? 5 seconds to answer. They are also termed as correct, cell-bound complement proteins. Okay, cell-bound or cell surface complement proteins because they are attached to the surface of your immune cells. They are designated by a descriptive title or letter. Example, decay accelerating factor. That's the example, no? DAF, decay accelerating factor. Or MIRL or MERL, membrane inhibitor of reactive lysis. The membrane-bound regulatory proteins have been assigned CD numbers, and descriptive titles and complement receptors are designated by numbers following the prefix CR. I actually placed in your notes a table summarizing the different CD numbers assigned for each regulatory proteins. What are the other names of regulatory proteins? Cell-bound complement proteins or cell surface complement protein. What is its function? It regulates or it inhibits the action of your or activation of your complement proteins. The other complement receptors are designated by the complement symbol followed by the uppercase letter R. For example, C2R. The R there would mean that that is a receptor. A receptor is a substance, no? A structure in the cell surface to which your complement proteins binds to. So C3R, diba? C4R. So what is the term for your C4 receptors? C4R. C3 receptors, C3R. So you just add the word or the letter, uppercase letter R, making it that, signifying that that is now the receptor for that specific complement component. So as I have mentioned in the previous slides, we actually have three ways to activate your complement system. That is via the classical pathway, the alternative, or via lectin pathway. Now, what I want you to remember on this slide is that the classical pathway begins with an antigen-antibody complex formation. Thus, it is considered as antibody dependent. Whereas your alternative pathway is initiated by cell surface molecules and does not need formation of antigen-antibody complexes. Thus, it is antibody independent. Your lectin pathway, on the other hand, only needs activation of your MBL, which binds to the mannose on the surface of your microorganisms, and does not need formation of antigen-antibody complex. Thus, this is considered as antibody independent. Now, the classical pathway is actually termed as classical because it was the first complement pathway to be discovered or to be identified. Please remember that because that came out in the board examination. Why is the classical pathway termed as classical or name called classical. A, because it was first identified, or B, there was classical music playing in the background during experimentation. So the correct answer was letter A. No? So it is an antibody-directed mechanism for triggering complement activation. Why antibody-directed? Because you cannot actually cause activation of your complement classical pathway if you do not have any antibodies binding, no, provided that these antibodies interact with the antigen. So there must be first and foremost interaction of your antigen and your antibody. And interaction of your antigen and antibody produces this what we call antigen and antibody complexes. Thus, a prerequisite for the activation of your classical pathway is formation of antigen and antibody immune complexes. Now, what are these antibodies that participate in the classical pathway? So we have two, IgM and IgG, of which IgM is the most efficient antibody to cause activation of your classical pathway. Why most efficient? Remember, you learned in your previous lectures that IgM basically looks like a pentameric. No, it's actually a pentameric structure, meaning it's actually a big immunoglobulin, and by being big, what's the advantage is that it has many binding sites. In other words, the antigen could actually bind to many sites in the IgM. You got that? Okay. Whereas your IgG molecules, being smaller molecule, has fewer number of binding sites compared to your IgM. That's why this IgM is considered most efficient of all the antibodies that causes activation of your classical pathway. Now, another reason is that being a large molecule, you only need one IgM to cause activation of the classical pathway. Whereas you need two IgG molecules, at least two IgG molecules for you to activate the classical component pathway. So, in order of efficacy, IgM is the most efficient, followed by the different subtypes of IgG molecules. So, IgM is followed by IgG3, IgG1, and then IgG2, with decreasing efficacy in activating your complement classical pathway. Now, I want to direct your attention. No, I want you to put 100% of your focus on this slide. It's because if you do not understand this slide, you will not understand the succeeding slides. Okay, read with me. Go. Complement activation can be divided into three main stages, each of which is dependent on the grouping of certain reactants as a unit. What are these stages? Recognition unit, activation unit, and membrane attack complex. Again, there are three main stages of your classical complement pathway activation. In other words, your classical pathway must have to undergo these three sequential stages for you to cause lysis of the cells, the microbial cells. Stage 1 or phase 1 is also called the recognition unit or the recognition phase. Stage 2 or phase 2 is also known as the activation unit or the activation phase. Whereas the MAC complex or the membrane attack complex is also known as phase 3 or stage 3. Next, question. How many proteins make up the classical complement pathway? You learned no 9. And these 9 proteins actually has a specific stage to which it becomes activated, to which it becomes participative of, no? For example, such that the recognition unit is comprised of C1 complex. The activation unit is comprised of your C4, C2, and C3. Your membrane attack complex is comprised of C5, C6, C7, C8, and C9. Please take note of that, because our discussion on classical pathway will revolve on these three main stages. Let's start first with the recognition unit. Okay, let's start with the recognition unit. Why was this called recognition unit? Okay, so again, the first complement protein to become activated is C1. C1 complex, to be complete. C1 complex makes up your recognition unit. So what happens in your recognition unit? Why is this term as recognition unit? I have pictures to show you on the later portion of my slide. So just bear with me, For now, I want you to look at the presentation here. Now, supposing this is the microbial surface. Remember, your complement classical pathway does not become activated if you do not have formation of antigen-antibody complexes. So again, antigen. And then you need an antibody to be attached to the surface of the antigen. Now, attachment of your antigen to antibody is what we call complement fixation. So what happens in complement fixation? There is attachment of your antibody to the antigen surface, forming an antigen-antibody immune complexes. And the antibodies that participate in complement fixation are termed as complement fixing antibodies. So what are these antibodies again? In order of efficacy: IgM, IgG3, IgG1, and then IgG2 being the least efficient. Okay? So I mentioned this already, the reaction sequence. And what is that reaction sequence? C1 becomes activated first, followed by C4, C2, C3, C5, C6, C7, C8, C9. Which of these components are part of the recognition unit? C1 complex. Which of these components are part of the activation unit? C4, C2, and C3. Which of these components are found in the MAC complex? C5, C6, C7, C8, and C9. So these are other substances which directly initiate the classical cascade. So as you can see, most of them are actually microorganisms, to which the antibodies become bound to. And binding of the antibodies to the antigen causes formation of your antigen-antibody immune complexes. Antigen-antibody immune complexes formation leads now to activation of your C1 complex. Okay. Let's discuss now C1 complex, the component of your recognition unit. I think you will appreciate more this one if I show you picture. The picture is here. Okay, so I'll explain this in detail. What do you see? You see blue colored circle with lines there. What I actually see is a bouquet with blossoms, with bouquet with 6 flowers, 6 blossoms. Let me explain that. Again, your C1 being a complex is actually comprised of subunits. Otherwise, you cannot call it a complex if it does not have any components or subunits. And how many subunits of C1 are there? 3, namely C1Q, C1R, and C1S. Again, C1Q, C1R, and C1S make up the C1 complex. Look at the structure, look at the diagram shown here. Now, the C1Q is actually comprised of polypeptides. Again, remember, I mentioned this already. Proteins are substances made up of polypeptides. So if you break down proteins, you get to have polypeptides. If you further break down polypeptides, you get amino acids. Diba? Okay. So your C1Q is comprised of polypeptides, namely A, B, and C. Polypeptide A, polypeptide B, polypeptide C. And each polypeptide is comprised of 2, no? So you have 2 polypeptide A, 2B, and 2 polypeptide C, resulting into 6. So I want to direct your attention to the bottom arrow here, labeled C1Q, and we call it actually the stalk. The C1Q stalk is actually a collagen, looks like a collagen tail. Now, if you extend, if you go higher, you go up, before you get to actually reach the top, you actually see a bent portion here. This portion, the bent portion, is actually comprised of alanine residues, alanine amino acid residues. And if you continue, you go higher, you end up with globular or circular heads. And how many globular heads do you see? 6, of course, because you have 6 stalks, and each stalk ends with 1 globular head. So you have a total of 6 globular heads. Listen. Now, I want to direct your attention to the arrows here pointing to C1R and C1S. Take note that the C1R component is actually a dimer, meaning for one subunit of C1R, you actually see 2 of them. One subunit is comprised of 2 molecules. That's why it's called a dimer. So it's a paired molecule. And how many paired C1R molecules do you see? 2 of them. So you have 2 dimers of C1R. This is 1, and this is another 1 dimer. The same with C1S. You also have 2 dimer molecules of C1S. So this is 1 dimer and another dimer. Listen, what is the importance? Why I need to point this out? Class, why do I have to spend so much time on explaining this? Because this is not nonsense. It has actually a role in the activation of your complement system. Otherwise, if you have defective C1 complex, your complement classical pathway will not be activated at all. It will just stop there in the immune antigen antibody complexes formation. Listen, what is the significance of that dimer molecules of your C1R and C1S? And why is it that the C1Q is made up of stalks and globular heads? Let's start first with your C1Q. In order, ha? Kay sequential man siya. In order. Let's start first with your C1Q. Remember, this is your surface of the microbe. Your antigen and antibody binds to the surface. Diba? Your antigen and antibody binds to the surface. From there, your C1 actually binds to the antibody, your C1. What particular component or subunit of the C1 binds to the antibody that is bound to the surface of this antigen? It is C1q. And what particular part of C1q binds to the antibody that is bound to the microbial surface? It is the globular heads. So it is the globular heads which participates in the complement cascade. Okay. So how many of those heads do you need for your complement activation cascade to go on? You need at least the 2 heads. So one head attached to another molecule, antibody molecule, and one head attached to the other part of the antibody molecule. So you need at least 2 heads. Now, when these heads become bound to the antibody, so if this is the antibody, this is the antibody, antibody. When the head now, that head that I am pointing here in the arrow, becomes bound to the antibody, what happens is that it causes or it induces mechanical stress. And that mechanical stress causes activation of your C1R and C1S. How? Okay, how? This is how. Okay. This is how. Your C1R and C1S is comprised of 2 domains. And what are these domains? The catalytic, also known as proteolytic domain, and interaction domain. Now, from the term we learned, no, that proteolytic or proteolysis is the one which causes activation or breakdown of the subsequent complement protein molecule, thus causing activation. So the dimer, the arrow pointed here, the dimer C1R and C1S, actually each component has 2 heads. Look at. So one is the catalytic domain and one is the interaction domain. The catalytic domain causes activation of the next complement protein to be activated, whereas the interaction domain is the domain which interacts with the other complement proteins. So your C1R and C1S has catalytic and interaction domains. And what happens when there are catalytic and interaction domains? When C1q globular heads become bound to the antibody, what happens is that it induces mechanical stress. And this mechanical stress causes activation of your C1R catalytic and interaction domain. And then this further causes activation of your C1S catalytic and interaction domain. Okay, so I hope you got that. Now, your C1q, I mentioned a while ago, what particular part of C1q binds to the antibody, the head or the stalk? The head, diba? The head of the C1q binds to a particular portion of the antibody. And you have learned that we have FC and FAB region of your antibodies. Now, what portion of the antibody do these C1q molecules bind? At the FC region, such that for your IgG, it binds now to the CH2FC region. For your IgM, it binds to the CH3FC region. So how do you memorize this? How many G's do you see in IgG? 2. So that's the CH2, 2 there. If you rotate M to the right, you will form number 3. Thus the CH3 there. That's how I memorized it. So your IgG at the CH2 region, IgM at the CH3 region. Take note. What do you observe here? What do you observe? The C1Q, the C1R, and C1S complexes are actually bound to each other. That's why we call it C1 complex, because they are bound to each other. They form a complex. And that is because of what ion? Calcium ions. So without calcium, you will actually see dissociation. No? You see separation of C1Q, C1R, and C1S. And then what happens? You will not have activation of your complement system. So in patients with hypocalcemia, you actually don't have much activation of your classical pathway. That's why these patients are actually prone to infection. No, in patients with hypocalcemia. So you need calcium ions for your C1R and C1S and C1Q to stick together, forming the C1 complex. So this is an electron micrograph of C1 complex molecule. So looks good, no? A stalk, a bouquet with 6 blossoms. So I mentioned in the previous slides that IgM is considered the most efficient, no? Whereas your IgG is second to your IgM. Why? Again, your IgM is a big structure and it has many binding sites, and you only need 1 molecule of IgM compared to your IgG, which is a smaller structure and you need at least 2 molecules. Now, I want to point out here that your IgM actually has 2 configurations, the staple and the planar configuration. The staple configuration is the configuration of the IgM that it assumes when it is bound to an antigen. Whereas when it is just freely floating in the circulation, it assumes the planar configuration. So let's apply this. Question: which of these IgM configuration causes activation of the classical complement pathway? Is it A, staple, B, planar, C, both, D, none of the above? What's your answer? Answer is staple. Why staple? Because you need the antibody to be bound to the antigen, forming now antibody-antigen complexes. And the antigen-antibody complexes will actually cause activation of your C1 complex, because in the first place, your classical pathway is actually an antibody dependent. Whereas for your IgG, we need 2 of these molecules. Take note, your IgA, IgE, and IgD cannot activate the classical pathway. So this is a picture showing you the planar and staple configuration of your IgM. So the leftmost portion is the planar configuration, whereas the bent position is the staple configuration, which is bound to the antigen. The topmost picture is the planar configuration, which is freely floating in the circulation, whereas the bent portion is the staple configuration, which participates in classical pathway activation. So I've mentioned this already, no? That you only need a single molecule of IgM to activate the classical pathway, whereas you need 2 IgG molecules to activate the classical pathway. So much cost-effective in IgM compared to your IgG molecules. And when C1q becomes activated, what happens when C1q becomes activated? The globular heads becomes bound to CH2 or CH3. What happens? It causes the activation of your catalytic and interaction domain of your C1R and C1S. So the C1R subunit is a beta globulin and it forms a dimer. I mentioned this already, class. No need to memorize these numbers. The numbers, no, molecular weight, it won't matter. That will not come out in the examination. I will not vex you on that, no. Whereas your C1S subunit is an alpha, alpha globulin, and it also forms a dimer. Same with your C1R subunit. So, as a review, let's review. Your C1q globular heads becomes bound to your Fc region of the antibody. Once bound to the Fc region of the antibody, what region in IgG? CH2. What region in IgM? CH3. Once bound to the Fc region, what happens? It causes mechanical stress. And this mechanical stress induces or opens up the active site on C1R, causing it to become activated. C1R, when activated, causes now cleavage or proteolysis or activation of your C1S. And that is now the end of your recognition unit. What heralds the end, no, of your recognition unit? Formation of activated C1S. So after activation of your C1S subunit of the C1 complex, what comes next is the activation of your C4. Remember that your C1S has two subunits or two domains. And what are these domains? Catalytic and interaction domain. The catalytic domain of the C1S causes cleavage or proteolysis of your C4. Thus, activation of your C4, there must be the word, no, activation. Otherwise, I'll really mark it wrong during your examination. Activation of C4 is considered the commencement or heralds the beginning of the activation unit, because C4 is the next complement component to be activated after the C1 complex. Thus, C4 is also termed as the first activation unit. So your C4 complement protein is the second most abundant complement protein. What is the most abundant complement protein? Your C3. Second only is your C4. And your C4 is comprised of three polypeptide chains, alpha, beta, gamma, joined by disulfide bonds. So again, I mentioned that the activated C1S would actually cause cleavage or proteolysis of your C4. And cleavage or proteolysis of your C4 component will actually produce how many fragments? I mentioned this already, 2 fragments. And what are these fragments? The A and the B fragment. The A fragment is called C4A, whereas the B fragment is called C4B. Which of these fragments is considered the larger fragment? Correct, C4B. B is the larger fragment. Which of these complement proteins is considered the smaller fragment? A, correct. C for A for smaller fragment. And which of these fragments now participates in the complement cascade? Correct, the larger fragment or C4B. And how about your C4A? What happens to your C4A? It is a byproduct of proteolytic cleavage of your C4. C4A is actually An anaphylatoxin. An anaphylatoxin is a small molecule that binds the receptors present on mast cells and basophils, which induces degranulation and release of inflammatory mediators, including histamine. Again, I mentioned this is a byproduct, and so it does not play any part in the cascade. What plays part in the cascade is the larger fragment, which is your C4b. Your C4b now, being the larger fragment, attaches itself to the bacterial surface. So once attached to the bacterial surface, it now causes activation of the next complement protein to participate in that cascade. And what is the next complement protein to be activated by C4b? Correct. Your C2. So C2, on the other hand now, once activated by your C4b, no? C4b now, when I say activated, meaning I mentioned this already, that protein has now an enzymatic activity. And the function of your enzyme is to actually cause proteolytic cleavage of that complement protein to become activated. So your C4b would actually cause what? It would actually cause cleavage or proteolytic cleavage of your C2, which is considered the second activation unit. C2 is associated with factor B found on chromosome 6 in the major histocompatibility complex. So I will be discussing this in the next lectures. And C4b only is able to cause proteolytic cleavage of C2 in the presence of magnesium ions. Ions. What ions must be present for C1 complex to become stable and not become dissociated? Correct. Calcium ions. What ions now must be present for C4b to cause activation or proteolytic cleavage of your C2? Magnesium ions. Take note. So, C2 in the presence of your C4b becomes now cleaved. In the presence of magnesium ions, becomes cleaved into smaller and larger fragment. What is the smaller fragment? This is the exception now. Remember, C2b. Which one is the larger fragment? C2a. Which participates in the cascade? C2a, which is the larger fragment. And so, C4b bound to the surface of the microbial cell causes now proteolytic cleavage of your C2, producing C2a and C2b. C2a being the larger fragment, being the fragment participating in the cascade, binds itself in the presence of magnesium ions with C4b, forming now a complex we call C4b2a. This is also known as C3 convertase. Again, C4b plus C2a forms a compound known as C3 convertase or C4b2a with a bar on top. Remember, I mentioned that in nomenclature. If you put a bar on top, that means that specific component of the complement has an enzymatic activity. So why is it called C3 convertase? I will answer that question in the next few slides. So let's discuss first C2a and C2b. Again, which participates in the cascade? The larger fragment, which is the C2a. So C2a is the fragment participating in the cascade, which causes cleavage of the next complement protein, your C3 and your C5. I will discuss that in detail in the next few slides. So This is what I've mentioned, no? C4b, C4b2A is the complex formed when C4b binds to C2A forming C4b2A, which is also known as C3 convertase. From the term itself, C3 convertase, its target complement protein is your C3. So your C3 convertase or C4b2A now is the complement protein which causes cleavage of the next complement protein to be activated, which is in this case C3. Right. So let's move on to C2b. So again, I mentioned this is the smaller fragment, and so this does not participate in the cleavage. And being the smaller fragment, it is actually a byproduct of your complement activation. Your C2b also increases capillary permeability, thus causing edema. And this is very labile and will have a half-life of 10 minutes. No? So what's important here is that your C2b actually causes increased capillary permeability and edema formation. So I mentioned that your C4b2A and your C3 convertase, or also known as your C3 convertase, is the enzymatic complex that actually acts upon C3 complement protein, thus causing now C3 to become cleaved, proteolytically cleaved into two parts or two fragments, also known as C3A and C3B. So your C3 is termed as the third activation unit, and this is also called or considered the central molecule of complement activation. Central molecule of complement activation. Why? Why the central molecule of complement activation? For some reasons. Number one, C3 comprises the first, or the most abundant complement protein in your serum. And so cleavage of your C3 molecule by your C3 convertase would actually produce many molecules. And so C3 cleavage or proteolysis actually is considered an amplification, major amplification process. Why amplification? Because if you cleave C3 molecules, because of its number, because of its volume, it is the most abundant, you actually produce many fragments of the C3. Whereas your C4, being the second most abundant complement protein, when you cleave it by the C1s complement protein, you also produce many molecules. That's why C4 activation is considered the first amplification process in the complement classical pathway activation, whereas your C3 activation or cleavage is considered the second amplification process in your complement protein classical pathway activation. So C3 is the major constituent. Why? Again, it's because it's the most abundant complement protein. And usually, it is consists of two polypeptides, alpha and beta, joined by disulfide bonds. And this is synthesized mainly in cells, particularly the hepatocyte. So C3 cleaved by your C3 convertase, also known as C4b2A. What are the fragments produced? Correct. C3A and C3B. Which of these fragments is the smaller fragment? C3A. Which of these fragment is the larger fragment? Correct. C3B. And which of these fragments now participates in the cascade, classical pathway cascade? Correct. The larger fragment, which is C3B. What happens to your C3A? C3A is a byproduct of your complement activation. Actually, C3A is C3A is an anaphylatoxin. Again, what is an anaphylatoxin? An anaphylatoxin is a small molecule which binds to the receptors of mast cells and basophils, causing degranulation reaction and release of inflammatory mediators and histamine. Alright. So your C3b is the component which actually causes the activation of the next complement protein. No? Also, another function of your C3b, this is also an opsonin. Opsonin. So again, what is an opsonin? These are actually substances which binds to the surface of your blood cell, of your cells, no, of your foreign cells, making it palatable or making it viable for opsonization. So I don't need to discuss what opsonization is because this was discussed previously in your innate immune system discussion or lecture. So what's important here, what I want you to note is that C4b, C2a, C4b2a or C3 convertase, when cleaving, your C3 forms C3a and C3b. Your C3a does not participate, it is just an anaphylatoxin. Whereas your C3b now participates in the cascade, and this C3b now binds to your C3 convertase, forming another complex which is now termed as C4b, 2a, 3b, which is also known as C5 convertase. Again, when C3 binds to your C3 convertase or C4b2a, it forms now another complex which is known as C4b2a3b, called also termed as C5 convertase. Now from the term itself, what complement protein does your C5 convertase target next? Very good. Your C5 complement protein. And no, the cleaving, once your C5 now becomes cleaved, or once it undergoes proteolytic cleavage or proteolysis, it forms now two fragments, your C5a and your C5b. Your C5a is the smaller A, no? A is the smaller fragment, whereas your C5b is the larger fragment. So which of these complement proteins now participate in the cascade? Very good. The larger fragment, which is your C5b. Formation of C5b and C5a is the end of your activation unit. And binding of your C5b into the cell membrane of the bacteria or to the cell membrane of the foreign molecule is the commencement or beginning of the formation of your membrane attack complex, which is considered stage 3 of your complement classical pathway activation. I hope you're getting that. So again, your C5a is an anaphylatoxin. An anaphylatoxin is a small molecule which binds the cell surface of basophils and mast cells, causing degranulation reaction, causing release now of inflammatory mediators and your histamine. Your C5b is the one participating in the cascade, and thus binding of your C5b to the membrane of the bacteria is now the beginning of your MAC complex. So your MAC complex again is comprised of the following complement proteins: C5b, C5, C6, C7, C8, and C9. Class, parang awan nyo na, huwag kayong sumagot sa exam ng C5 kapag tinanong, what are the components of the membrane attack complex? Why? It's because your C5 is still part of your activation unit. But once your C5 is cleaved into C5a and C5b, that is now the beginning of your membrane attack complex. Ha? Take note. So what happens in your membrane attack complex? Your C5b attaches itself into the surface of the bacteria. C5b is still unable to cause lysis of the cells. Thus, it has to recruit another complement proteins, which in this case are as follows: C6 and C7. And once your C6 and C7 binds to your C5b, it forms now the complex C5b 6 and 7, which does not still cause lysis. It needs another molecule to cause lysis. Now, binding of the next complement protein C8 to that complex C5b 6 and 7 causes now insertion of the C5b 6, 7, and 8 into the membrane surface, causing now small lysis. Small lysis, no? Why small lysis? Because you actually need another complement protein to complete the lysis. Class, parang ganito. Kapag nagpukpok kayo ng nail, no, a nail, when you hammer a nail into a wood, you actually have to hammer, pound, no, many times. And that last pound causing the nail to be really subconjugated, no, malubong siya all throughout the wood, is actually when that last hammering happens. That is just the same with your MAC complex formation. When C5b6, 7, and 8 forms a complex now, there is small lysis in just enough to lyse non-nucleated cells, no, cells without nucleus. For example, your red blood cells. But when C9 now binds to your C5b 6, 7, and 8, this causes now complete lysis of your nucleated cells, even non-nucleated cells. And so the complete membrane attack complex is C5b 6, 7, 8, and 9. So I hope you're getting that. Huh? So when C9 attaches to the initial complex, this forms pores, the pores that you have seen in the first slide that I have showed you. And pore formation would actually cause influx of ions followed by water and swelling and then rupture by osmotic lysis. So what happens is that what's the major cation inside the cell? Potassium. So what's the major cation outside the cell? Sodium. So once there is pore formation or hole formation on the surface, sodium gets in, followed by water, and then your potassium goes out. Eventually, since more and more water goes in, what happens is that your cells now becomes bloated and then eventually burst or lyses. So gram-negative are particularly susceptible to destruction by the MAC complex, whereas the gram-positive bacteria, which has intact cell wall, resist lysis and instead are targeted by phagocytes following deposition of your C3b on the cell surface. So let me explain this one by one. Why is gram-negative more susceptible to your MAC complex? Remember, your gram-positive cells, the surface of your gram-positive cells are actually comprised of lipid bilayer. So by lipid bilayer, it would actually mean that on the surface, below the surface, you actually see lipids, no, or hydrophobic layer. Meaning these layers are actually afraid of water, and so they actually hide themselves from the surface, no, from watery surface. And your MAC complex is also nonpolar or hydrophobic. And so in physics, we learned that like attracts like, opposite repels. No? So what happens? Your lipid bilayer being hydrophobic and your MAC complex being hydrophobic would actually cause attraction to each other, and so they do not repel normally. And so what happens there is that it would be easier for your MAC complex to insert into the lipid bilayer compared to the gram-positive bacteria, which the cell membrane surface is comprised of an intact cell wall. So much difficult for the MAC complex to penetrate. That's why gram-positive bacteria are actually more targeted by deposition of your opsonins. So what complement protein is considered an opsonin? C3b. Correct. Very good. So to wrap up today's lecture, I want to end this lecture, part 1, with these pictures. No? So I actually invited my friend here, a bacterium. I actually invited a bacterium for you to show to show us what will happen when that bacterium is attacked by your complement system. So please be interactive. Please answer my questions, no, as we go on. So again, you need to use more of your senses for you to learn better. Okay? So bacterium. Once there is bacteria detected by your immune system, what happens next before your complement classical pathway becomes activated? Very good. What happens is that there is recruitment of your complement fixing antibodies. And we call now the complement fixing antibodies. Say, complement fixing antibodies, come on down. All right. So these are your complement fixing antibodies. Now, that alone, complement antibodies, fixing antibodies, plus your antigens found in the surface of the bacterial cell wall, will that cause activation of your complement classical pathway? Yes or no? No. Why? We need the antibody to be bound to the surface of the cell membrane of the bacteria to form an immune antigen antibody complex. What do you call that process of binding of the antibody to the surface of the bacteria? Very good. Complement fixation. Now say, bind. Of course, the antibodies now are bound to the surface of the bacteria. Can this cause now activation of your complement classical pathway? Yes, very good. And what complement protein becomes activated first, or what complement protein in the classical pathway participates in the cascade? Correct. C1 complex. There must be the word complex, otherwise I'll mark it wrong. C1 complex. Can you call C1 complex for me? Say C1 complex. Come out now. There you go. C1 complex now is ready and primed to participate in the classical pathway of your complement system. Now, what happens to your C1? C1 being a tri-molecular subunit. No, it has 3 subunits, your C1Q, your C1R, and your C1S. Your C1Q is comprised of 6 stalks ending in 6 globular heads. How many globular heads needs to bind to the FC region of the antibodies for it to become, or for it to cause activation of your C1R and your C1S? How many? Correct. 2. Can you say now bind for C1Q to bind to this FC region? Say bind. Bind is now bound. Okay? So, question. What particular FC region does the C1Q globular head bind to if the antibody is IgG? Very good. You're actually listening to my lecture. CH2 region. How about if IgM? CH3 region. And what particular configuration of IgM now causes activation of your complement protein? Very good. Planar or pentameric configuration. Okay. So C1q globular heads now are bound to your complement fixing antibodies. What happens next? This induces mechanical stress to the C1q heads and stalk, thus causing activation of your C1R. An activation of your C1R now causes activation of your C1S. How many domains of C1R and C1S are activated? 2. We have the catalytic and interactive domain. Say, become activated. Yes, now, they are now activated, your C1R and C1S, and so they are now ready and primed to activate the next complement protein. Question: what specific subunit of the C1 complex causes activation of the next complement protein, such that activation of this complement protein heralds the beginning or commencement of your activation phase? Correct. Your C1S. Now, what complement protein is activated after C1S is activated? Very good. Your C4 subunit. So can you call C4 now? Say C4. C4 is here. C4 becomes activated by your C1S now. Say activated. It is activated into two fragments, producing C4A and C4B. Which of these fragments now participates further in the cascade? C4B. Which is the larger fragment? C4B. C4A being the smaller fragment is actually a byproduct of your activation of the classical pathway. C4A is considered an anaphylatoxin. An anaphylatoxin is a small molecule which binds to surface receptors of your histamine and basophil, triggering degranulation and release of immune mediators or immune substances and release of your histamine. What happens now to your C4B? C4B being the larger fragment participates in the cascade. So we have now C4B. Your C4B must be attached to the surface of your bacterial wall for it to cause activation of the next complement proteins. What is the next complement protein to be activated by your C4B? Correct. Your C2. Can you call now? C2. Say C2. Very good. C2 is ready to participate now in the cascade. Is it? Not yet. It's because your C2 must be activated first by your C4B component for it to be able to participate in the cascade. And so C2 now becomes activated and is cleaved by your C4B into how many fragments? Correct. 2 fragments. Say cleaved. Cleaved. Okay. So it produces now C2A and C2B. Binding of your C2 fragment to C4B happens only in the presence of what ions? Very good. Your magnesium ions. So the absence of your magnesium ions would actually cause dissociation or would not cause binding of your C2 molecules. Now, once bound, it becomes now cleaved into C2A and C2B, which is the larger fragment. So this is the exception, no? C2A, which is the smaller fragment. C2B. Always, it is the larger fragment which participates in the cascade. So therefore, your C2A now being the larger fragment participates in the cascade, and once bound to your C4B forms a complex called as C4B2A, also known as your C3 convertase. Your C3 convertase now is an active enzyme which targets what? Complement protein. Very good, your C3. Call C3. C3. C4B, C2A, or C3 convertase targets C3. Now C3, for it to become activated, has to be cleaved by your C3 convertase or C4B2A molecule. And so, cleavage of your C3 would actually produce fragments. So you say, cleaved, C3, be cleaved now. C3 is cleaved now into two fragments, C3A and C3B. Which of these now participates in the cascade? The larger fragment, which is your C3B. Whereas your C3A being the smaller fragment is an anaphylatoxin, a byproduct of your classical complement pathway activation. What is an anaphylatoxin? An anaphylatoxin is a small molecule which binds to the cell surface of your mast cells and basophils, triggering release or degranulation reaction, causing release of your immune mediators, inflammatory mediators, and release of your histamine. Now, binding of your C3B to C4A2B or C4B2A, C4B2A, also known as your C3 convertase, produces another complex, enzymatic complex. And what do you call that enzymatic complex? Very good, class, for your answer. C5 convertase, also known as your C4B, C2A, C3B, or C4B, 2A, 3B. From the name itself, what complement protein does your C5 convertase targets? Very good. Very good. Your C5. So can you call C5 for me so that our complement cascade will continue? Say, C5, come on down. C5 is now ready and able to participate on the cascade. So again, remember, class, what happens is that your C3 convertase, your C5 convertase does not only cleave one C3 and one C5 molecule. Again, your C3 convertase and your C5 convertase does not cleave only one C3 and C5 molecule because you do not have only one C3 in your circulation. In fact, C3 is the most abundant, diba? So it cleaves many molecules, and so you produce many complexes, many byproducts also. So your C5 convertase causes cleavage of your C5 forming C5A and C5B. Which of these complement proteins participates in the cascade? Very good. The larger fragment, which is your C5B. C5A, on the other hand, being the smaller fragment, is considered an anaphylatoxin. An anaphylatoxin is a small molecule which binds to the surface receptors on mast cells and basophils, causing degranulation and release of immune inflammatory mediators and your histamine. So we have now C5 being cleaved into C5A and C5B. C5B is the one which participates in the cascade. C5A is just a byproduct. So your C5B, what happens next? For us to form MAC complex, it has to be bound to the surface of the bacterial wall. Otherwise, it will be it will be destroyed, no, in the circulation if it does not become bound to the surface of your bacterial wall. So say bound. Bind C5. Bind C5. C5 now, together with your trimeric complex C6 and C7, becomes bound to each other and binds themselves into the, into the cell membrane layer. Question, does this cause pore formation? No. We need another complement molecule for us to cause lysis of your cells. And what is that complement molecule that becomes activated next? Very good, your C8. Can you call now your C8? Say C8. C8 now is ready to participate in the cascade. What happens to C8 is that it binds itself into the trimeric complex of C5b, 6, and 7, forming now another complex C5b, 6, 7, and 8. Can this cause lysis of cells? Yes. What cells? Cells without nucleus or non-nucleated cells. But we need another molecule that would actually cause complete lysis of your bacterial cell or foreign cell. And what is that complement molecule? C9. Very good. So can you call C9 for me? Say C9. C9 is ready and primed to participate in the cascade. And you need at least 12 to 15 molecules of C9. Actually, in some books, no, 1 to 18. In some books, 12 to 15. So you follow my notes, what is written in my notes. So you need a lot of C9 molecules to actually cause pore formation. So what happens to C9 is that it forms a hole in the membrane of the bacterial wall. So it undergoes polymerization. So you need more and more and more of your C9 molecules to form a hole in the membrane surface. And once there is a pore now, or hole in the membrane surface, what happens? Ions from the outside goes inside together with your water molecules. And ions from the inside goes out from your cell membrane surface, causing now leakage. So ions leak out and thus eventually causing lysis of your bacterial cell. So that is now how your complement classical pathway becomes activated and causes lysis of your cells or foreign cells. Thank you everyone for your kind attention. Please stand by for part 2 of my lecture. I will be discussing the alternative pathway, the MBL pathway, and some diseases that happens when there is unregulated activation of your complement system. Thank you guys. Study hard.